Florian. It's great to speak with you today. As 10x Genomics are sponsoring Spatial Biology Europe Online, I thought it would be great to catch up with you. What are your thoughts on the current state of spatial biology?
I'm really excited about spatial biology. The field is really exploding. And seeing all those measurements on cells in their natural context, which is tissues, really allows us to gain a completely new understanding of biological systems. So I'm really keen to see what's coming.
What issues does spatial biology address?
I think we have a good understanding, or we have an increasingly good understanding of single cells and can do a lot of measurements. But in order to actually really understand the tissues and the tissue structures where cells are very often located in nature, we also need that spatial information. So combining this is really powerful because if you, for example, look at developmental biology, we really understand there's very often different layers of developing tissues that have different functions. So we really want to understand them spatially in order to understand their function even better.
And where does 10x fit into this ecosystem?
At 10x, we respect that biology is very complex. Our mission is to help researchers resolve biology to advance human health. For this, we have a lot of different tools who allow to do measurements on different modalities, such as, for example, RNA and protein. Our Visium platform especially provides really a complete tissue overview of your tissue of interest. You have up to 5,000 spots that measure the transcriptome in your tissue section, and you get an image of the tissue as well. And this is really the perfect discovery tool where you can investigate the tissue, the morphology, based on information that we've had and experience that we've had for a long time, for example, in pathology especially. And then you can add on gene information, so gene expression information, where you can understand what are, for example, driving genes for a specific morphology. And if we come from very often more low-plex technologies, maybe it's just such as LCM, so where you cut out a few parts of the tissue or other of these low-plex technologies, which force you to kind of select the region before. But what you can do with the Visium is that you capture the entire transcriptome from the tissue section, and then in analysis, you can select those regions of interest. So you capture for one section everything that there's to know from the transcriptome, and then you can actually design your experiments afterwards. So you can do an experiment, and then you can redo it, and so on, as many times as you like. So I think this is really the perfect tool for doing unbiased discovery.
What can participants expect from your workshop at Spatial Biology Europe Online?
As Visium combines histology and gene expression in a totally new way, we've also seen the necessity for groups working together to really have all the know-how that they need to run this assay. So at the Newcastle University, we've seen, for example, the pathology, the genomics, and the bioinformatics department collaborate together to create Visium data and actually offer that as a service. And so we have them present how they achieved this. And then afterwards, there will also be a panel discussion that will give you the chance to ask any questions you might have from people who are actually using the technology hands-on.
That's great. Thank you very much for your time today, Florian.